With advancements in genetics, molecular biology, and tissue culture, new traits have been incorporated into crop plants. Explain the main steps in breeding a new genetic variety of a crop. So, the main steps in breeding a new genetic variety of a crop are: there are 5 steps: collection of variability, evaluation and selection of parents, cross hybridization among the selected parents, selection and testing of superior recombinants, testing, release, and commercialization of new cultivars. So, coming to the collection of variability. So, here the collection and preservation of all the different wild varieties, species, and relatives of the cultivated species is a prerequisite for effective exploitation of natural genes that are available in the population. So, basically, here the entire collection of plants and seeds having all the diverse alleles for genes in a crop is called germplasm collection. So basically, they just collect all the plants and seeds having all the diverse alleles for the gene. Next step is evaluation and selection of parents. So the germplasm that is evaluated so as to identify plants with desirable combination of characters. So basically, in the next step, in this step, germplasm is evaluated. So the selected plants are then multiplied and then used in the process of hybridization. So basically, in the second step, pure lines are created. So next, the third step is cross hybridization among the selected parents. So here the desired characters have very often to be combined from 2 different plants or say parents. See, for example, one plant has high protein quality that may need to combine with disease resistant from another plant. So this is possible by cross hybridizing the 2 plants to produce hybrids that genetically combine the desired characters in one plant. Fourth step, selection and testing of superior recombinants. So this step yields plants that are superior to both of the parents. So this step consists of selecting among the progeny of the hybrids those plants that have the desired character combination. So these are self-pollinated for several generations till they reach a state of uniformity. It means till they reach homozygosity, they will be self-pollinated so that these characters will not segregate in the progeny. Final step is testing, release, commercialization of new cultivars. So the newly selected lines are evaluated for their yield and other agronomic traits of quality, disease, resistance. So once the varieties are produced, so we need to check for their quality, disease, resistant. So this evaluation is done by growing these plants in the research fields and recording their performance under ideal fertilizer application, irrigation, and other crop management practices. So the evaluation in research field is followed by testing the materials in farmers field for at least 3 growing seasons at several locations in the country, so that we will come to know the representing all the agroclimatic zones where the crop is usually grown. We will come to know what is the quality of these new cultivars. So the material is evaluated in comparison to the best available local crop cultivar, a check or reference cultivar. So these are the main steps that are involved in breeding a new genetic variety of a crop.